Hello, my name is Wang Ziyan. I'm from the University of Hong Kong, Department of Diagnostic Radiology. I'd like to share our recent publication, Saturation Transfer-Based MRI of the brain in multiple sclerosis patients S3T. Multiple sclerosis is an autoimmune demyelinating disease. The immune system attacks myelin, which helps the transmission of electrical signals in the brain. And this disease will cause demyelination and neurodegeneration. These damages are irreversible. MS is also one of the main causes of non-traumatic disability among young adults. In this study, we aim to propose a saturation transfer-based MRI protocol that includes CEST MRI for assessing molecular changes and MISL MRI for evaluating water exchange in the brains of patients with MS at clinical 3T. The first part is CEST MRI. All MRI scans were performed on a GE 3T scanner. We used 3D CUBE sequence with 12 slices and 6 mm thickness. CEST employed a continuous wave saturation pulse with a power of 0.8 μt and the duration of 2 seconds. Other parameters are also listed here. This study included 30 healthy controls and 21 MS patients. As we can see from the table, there is no significant difference between two groups. CEST MRI can detect multiple endogenous molecules with exchange protons, as we can see from this illustration. The 3T study mainly focuses on the MET at 3.5 ppm for measuring proteins and peptides. The CEST signal at 2 ppm that is primarily contributed by creatine. The RNE at -3.5 ppm highlights mobile lipids and proteins, and the magnetization transfer contrast at -2.5 ppm reflects signals from semi-solid macromolecules. Two analysis methods were used here, including our recently developed double-step multipool Lorentzian fitting and Lorentzian difference analysis. Here are the results. This slide shows the comparison of a brain slice of T1 weighted image, T2 weighted image, and CEST maps for a healthy participant and an MS patient. A decrease in the CEST contrast was observed in all CEST maps of the MS patient, especially RNOE and MT. Also, an enlarged ventricle region caused by brain atrophy, indicated by a white arrow, was clearly observed in the MS patient images. This figure shows the group comparison of all CEST signals generated by two methods. We can see except CEST at 2 ppm extracted by DMPRF, all other CEST contrasts show significantly lower signal in MS group compared to HC group. MS signal drop in MS could be due to the immune-mediated inflammatory responses that destroy myelin and cell structure. And the signal reduction in ROE and MT may be contributed by compromised lipid integrity and loss of other macromolecules in the brains of MS patients. The results of ROC analysis show that, except for CEST at 2 ppm, all other CEST signals exhibited an AUC are greater than or equal to 0.88 in differentiating MS from HC. Furthermore, we also perform ROC analysis for the combined CEST signal for each method. We can see that DMPLF shows a higher performance in differentiating MS from HC with an AUC of 0.93. We also studied CEST signal changes in MS lesions. Images from a representative MS patient are shown in this slide. This patient had demyelinating lesions, indicated by the white arrow. The lesion area shows hyperintensity in T1 image, hyperintensity in T2 image, while in CEST maps it shows low signal. This result is consistent with our previous RNE study, as shown in the figure on the right. where the lesions are circled. These results provide additional insights for the diagnosis for MS lesions. The second part is MISL MRI. Before talking about MISL MRI, I would like to briefly introduce glymphatic system, as MISL detects CSF tissue water exchange in glymphatic system. As shown on the left illustration, the glymphatic system is a brain-wide pathway along a system of connecting perivascular spaces. On the right figure, we can see the glymphatic system maintains brain homeostasis by promoting the water exchange between CSF and brain tissue, which helps remove metabolic waste. MISL MRI is a relatively new saturation transfer-based MRI technique. It can quantify water exchange between CSF and brain tissue by selectively labeling water molecules in tissues through a magnetization transfer process. After that, the labeled water molecules will be transferred to CSF via CSF tissue water exchange and cause a water signal reduction. Previous studies confirmed that MISL MRI can detect age-dependent CSF tissue water exchange and reduced water exchange caused by AQP4 suppressors, but the application of MISL MRI in the neurological diseases remains unexplored. In MS brains, the inflammatory responses may cause an alteration in CSF tissue water exchange. MISL MRI scans were performed on the same 3T-GE scanner. For echo time optimization, TE was varied at 60, 100, 140, and 180 milliseconds, with a saturation power of 2 μT and a duration of 2 seconds. After optimization, TE was set as 180 milliseconds for MISL acquisition. Reference image at -300 ppm, labeling images at -20 ppm and -10 ppm were acquired. In MISL study, we finally included 14 healthy controls and 12 MS patients for group comparison. In TE optimization, both CSF and brain tissue show intensity decrease with increasing TE, with a significant drop after 100 milliseconds. Notably, brain tissue decayed at a faster speed compared to CSF. To be more specific, at 140 milliseconds, brain tissue intensity drops by 31.53% and CSF intensity by 8.74%. At 180 milliseconds, brain tissue intensity drops by 50.39% and CSF intensity by 14.46%. Hence, a TE of 180 milliseconds was chosen for the formal MISL acquisition as it offers best contrast between CSF and tissue. The comparisons of MISL signals between HC and MS are shown in this figure. The intracranial MISL maps of MS patients exhibited lower signals, especially in the area of subarachnoid space. An MS group showed significantly lower MISL signals than HC at both -20 ppm and -10 ppm, indicating a slowdown in CSF tissue water exchange in the brains of MS patients. In summary, we found that MRI can effectively detect molecular changes in MS brains, with DMPRF exhibiting an improved diagnostic performance with AUC of 0.93. MISL MRI reveals decreased CSF tissue water exchange in MS patients' brains. There are also some limitations, such as the long scan time, large slide thickness, and limited sample size, which should be addressed in future large cohort studies. Overall, our results highlight the clinical translational potential of saturation transfer-based MRI, providing additional insights for the diagnosis of multiple sclerosis. Thank you for listening, and thanks to all lab members and collaborators.